Today's event, just a couple of logistics, is going to be recorded and later made available through the YouTube channel of the Royal Society of Edinburgh. All the attendees can submit questions at any time via the Q&A tab at the bottom of your screen. There is also going to be a feedback questionnaire that is going to be distributed to all the participants so that we can actually capture some of the feedback, how you felt about it, and we can maybe improve that in the future. And during the event, feel free to use this hashtag here to actually live tweet during the discussions. So just a very brief overview of the event. So it is split in two sessions. So part one, we're going to discuss about the scientific aspects using interdisciplinary approaches, so combining medicine, biology, chemistry, physics, biophysics, data science, and omics approaches, as well as physiology. And we'll have also the last speaker giving a perspective from an early career researcher to mitochondrial research. Then we're going to have a short Q&A session, and then we will break for 10 minutes just to stretch out a bit. And then we're going to move on to the second session, which is the societal issues, and we're going to discuss on the experiences of patients and patient group organizations, bioethics, sociology. We're going to discuss about an activity which is really important, the involvement of women in mitochondrial research and a network that has been recently organized. And also we're going to bring into the discussion the designers and how they view innovative approaches, how they can organize innovative approaches for engaging with larger audiences. We will then have a Q&A session for that second part, and then I will close at 3 o'clock with some final remarks. So we have a very good selection of speakers coming from different disciplines. In the first part on the science, what do we know about mitochondria? Where do we go from here in terms of advances in mitochondrial research? Bob Lightowlers from Newcastle, Gloria, Brea, Calvo from Seville, Richard Hartley from Glasgow, Stefan Jakobs from Göttingen, Hana Antonicka from McGill University in Montreal in Canada, and Bruno Chausse from Heidelberg. And in the second session, we'll have the impact of society. My colleague Simon Woods will chair that session after the break. So just very, very briefly to introduce the mitochondria. Of course, people participating in this event either come from a scientific background or more from a societal background. So just to set the scene about mitochondria, so these are organelles that are inside the cell. They are known with this very generic term as powerhouses of our cells. The name, which is sometimes very difficult when I teach students, there's always a question about what is a mitochondrion and what are the mitochondria. So this comes from the Greek, so I mean an advantage to actually understand and explain that. The term was coined in 1898 by Carl Brenda, and we can see here these beautiful electron tomography slides of these organelles in the cell. They're not just powerhouses, so they're not just producing energy, but during the talks we're going to hear about other things that mitochondria are involved with. And the recent term that I saw, and I think is very appropriate on Twitter using for mitochondria, is that they function actually as the CEO, which stands for Chief Executive Organelle of the cell. They control not just normal cell physiology and metabolism, but also cell death. So they have an absolutely vital role in many of the cell functions and cell development and also cell death. So to start off with mitochondria, they are made following a very elaborate process. So already to make mitochondria, it's a rather complex process, and we know a lot about it. And what is really important in this is that this is the combination of two genomes, of the expression of two genomes, not just the nuclear genome, but also the mitochondrial genome. So this complex process of mitochondrial biogenesis mirrors also the complexity of the function of mitochondria. But when mitochondria dysfunction, this is really crucial for the cell. So they are linked to several of the common human diseases, like cancer, neurodegeneration, and also diabetes. And also they are involved and they modulate the impact of infections, be it bacterial or viral infections like COVID, for example. Primary mitochondrial disease is a devastating, these are devastating disorders, and they are sadly incurable. I think in the first talk, then I will pass this now on to my colleague Bob Lightowlers. We will hear a little bit about, where we stand with in terms of mitochondrial disease, and I'm sure we'll have some discussion about potential mitochondrial therapies. In mitochondrial research that has spanned decades, there have been several seminal contributions. Many of them are very well known and recognized. For example, here, the two very well-known Nobel Prizes for Peter Mitchell for the chemiosmotic hypothesis, and later on to John Walker for the structural analysis of how ATP is generated through the ATP synthase. And we're lucky in the sense that in this field, we have had all these very major figures contributing to the absolutely fundamentals of how mitochondria work. And now we can make progress in terms of understanding mitochondrial dysfunction and link that to the molecular architecture and basic mechanisms of mitochondria. So mitochondria research is advancing at a very rapid pace. It is a very exciting field, and it is also hugely interdisciplinary. You will hear talks from people coming from chemistry, physics, omics approaches, medicine, etc. And it is also very international, and that is reflected in the panels, in the background of the people, and this is also something we're going to discuss at the end. So without any further ado, I would like to pass it on to Bob Lightowlers, who is the first speaker of the session. I will now stop sharing my screen, and I will ask Bob to take over. Bob, if you can just start your talk, please.
Hopefully everyone can hear that. Thank you very much, Kostas. And I'd just like to start by saying a big thank you to the Lilly Foundation, and also to the many patients who collaborate a lot with different people in the Wellcome Centre, which I'm part of in Newcastle University. So what I'm trying to show by this first slide is that mitochondrial disease is very difficult, notoriously difficult for clinicians to spot and to diagnose. They can present at any stage of life, and they can be extremely profound, as Kostas was saying, or they can actually be very, very, they have only a minimal clinical effect. So they are notoriously tough. Not only that, they present in very different ways, and very different tissues are affected. And often you will see neurological aspects of disorders, but there's also a lot of non-neurological association and affected tissues as well. Now here I've focused on the mitochondrial genome, but as I'm sure you may be aware that mutations or pathogenic mutations may be found in the mitochondrial genome, but they can also be found in nuclear genes. And in fact, at the moment, we know of more than 300 different genes that have mutations which can cause disease in mitochondria. How common are these disorders? Well, the minimal point prevalence for mitochondrial DNA mutations is about 1 in 5,000. And with respect to those nuclear genes, which I just mentioned, overt disease is due to around about 3 in 100,000. So we can say the prevalence is around about 1 in 4,300 patients, which for genetic disorders is really quite common. Treating the treatable. Now, as Kostas has said, unfortunately, there is no current therapy, curative therapies that we can offer, but clinicians can offer to treat a whole variety of different presentations of those disorders. And I list those here, but perhaps perhaps most commonly, say, diabetes, it might be possible to use insulin, of course. And for cardiac problems, we might be able to offer heart pacemakers. However, there are no curative treatments for these, what are often very profound mitochondrial diseases. So over the last 5 or 10 years, scientists around the world have been really coming together to try and address this problem, and whether or not there is any possibilities at all of developing therapies for mitochondrial disease. Now, I've listed a few here, and of course, I don't really have enough time to go into details, but if you would like to ask me any questions in relation to any of these disorders, or just to developing therapies in general for these disorders, please do either just send those questions through to us. So just very briefly, if we look here at this list, we can see promoting mitochondrial biogenesis. We're very interested in identifying small molecules that could do this. There's a lot of interest in eliminating pathogenic mutations through genetic approaches at the moment, and there's a whole variety of other different approaches. And just finally, for my little session, I'd just like to emphasize the key challenges that we see in mitochondrial disease: identifying novel therapeutics, understanding tissue-specific presentations of those disorders, and trying to determine genetic cause underlying late-onset dominant disease. So that's a very rapid twirl and whirl around the world of mitochondrial disease and therapeutics, but I hope you'll find you have many questions, and feel free to send them through to me. Thank you very much, and I'll hand that back now to Kostas.
Thank you very much, Bob. Appreciate that, and also staying on time. So we need to move on and keep track of time. So we move on to the next speaker, Dr. Gloria Brea Calvo from the University in Sevilla in Spain. And Gloria is going to talk to us about assembly of coenzyme Q in mitochondria, one of the important small molecules. And I will pass it now to Gloria.
Okay, thank you. Hello, everyone. I will firstly like to thank you for inviting me to this super interesting event, and I'm looking forward to continue hearing the rest of the speakers and also to the Q&A session. Well, we are, as Kostas said before, we are interested in coenzyme Q at different levels. Coenzyme Q is a small lipid that is present virtually in all cell membranes, but specifically also in mitochondria. You may know about coenzyme Q because, well, it is also called the Q10. So you may know coenzyme Q by the cosmetics, but I can tell you that this is a molecule way more important than just an additive of an anti-aging lotion. In fact, coenzyme Q participates as an essential element of the respiratory chain, so eventually allowing the synthesis of ATP. It is made up of a head with redox activity and also a very long tail that anchors the molecule to the inner membrane of the mitochondria. If we make a little bit of a zoom, we can see how it is integrated in the inner mitochondrial membrane, and there it transfers electrons from complexes 1 or 2 to complex 3. But not only, it also receives electrons from other enzymes, so it's participating in many other processes. For example, the synthesis of some types of nucleotides, actually. And where does it come from? Coenzyme Q can be incorporated by the diet, but it is actually mainly synthesized inside the cell, actually in the mitochondria mainly. And it depends on a set of nuclear encoded proteins that are working probably as a complex attached to the mitochondrial inner membrane, as I said, in places that are close to the ER mitochondrial contact sites. Well, the head comes from a very unknown pathway, from probably an amino acid, and the tail comes from a pathway that is shared with the other molecules, synthesis, such as the cholesterol, which is very interesting, but I don't have time to stop here to explain that. But once the head and the tail are linked together by COQ2, the head suffers a sequential modification that are performed by the rest of the COQ proteins to get the final molecule. This is a very complex process that needs to be coordinated with the synthesis of the rest of the respiratory chain elements, which is quite unknown, this coordination. We recently have observed that cells that are lacking COQ6 or are mutated in COQ6 can benefit from treatment of vanillic acid that will restore the synthesis of coenzyme Q, which opens the door for new treatments of this type of diseases. Primary coenzyme Q deficiencies actually are genetic conditions that are caused by mutations in any of the genes involved in coenzyme Q biosynthesis. And when a patient is suspected to have a primary coenzyme Q deficiency, is normally subjected to genome analysis, and there are several options that can happen. So we can identify an already reported pathogenic variant, but we can also get an unknown pathogenic or an unknown variant that could be causing the disease. So what we do in our lab is to contribute to the diagnosis by functional studies, and we do basically two types of studies: biochemical characterization, measuring coenzyme Q levels or coenzyme Q biosynthesis rate, but also some phenotypic rescue assays. So we work for speed up the process of diagnosis of patients with primary coenzyme Q deficiency, because they can benefit, some of them can benefit with coenzyme Q supplementation. So we try to speed up the process. Thank you.
Thank you very much, Gloria. We have to move on. The next speaker is Richard Hartley from Glasgow. And Richard is going to talk to us about even smaller molecules and chemical probes that can be used, and see how chemistry can provide us tools to interrogate mitochondria function and potentially intervene with therapy. Richard, to you now.
So it's a great pleasure to talk to you about the mitochondria from a chemistry perspective. Now let's go back to basics. We all think of mitochondria as the powerhouses of the cell. That is, they take the fuel in the form of food, and it's broken down to metabolites that are used within the mitochondrial matrix to drive the power formation, that is the formation of ATP in the inner mitochondrial membrane. While this happens, one of the things that also is taking place is that there is this byproduct reactive oxygen species, and I'll come back to those in a minute. Now here what we have is a powerhouse, but a powerhouse is not very useful unless it has some control. And of course, we expect control to arise from the nucleus, and you've already heard that many of the components of this powerhouse are provided from nuclear-coded proteins manufactured in the cytosol. So there's communication from the nucleus, both in terms of proteins and other signals. But a useful powerhouse also communicates with the controller. And the mitochondria do this through their metabolites and through the reactive oxygen species. They tell the state of the mitochondrial, of how the mitochondria is it functioning well? It controls its fuel supply and its output, and it sets it in the correct perspective for what the cell as a whole is doing. So this is a smart powerhouse. It's interacting, it's contributing to signaling. Now, when things go wrong, there are three significant areas where you could go wrong. You could fail in the signaling. You could give an inappropriate signal to the whole cell. You could, in the case of these reactive oxygen species, cause direct damage, and that, in its most extreme case, will kill the cell. Or your power could fail. There could be insufficient ATP produced. So let's look at two of these. Let's look at signal failure. So in some cases, in some inflammatory diseases, you will see that this signal coming from the mitochondria in form of itaconate is not strong enough. And the result is that the inflammatory macrophages remain on. What can we do about that as chemists? Well, what we can do is we can deliver a cell-permeable version of the metabolite that's not getting through and switch on the signaling again, and that gives you the anti-inflammatory effect and switches off the macrophages. But that's acting on the signaling out with the mitochondria. Now let's look at the mitochondria in detail. Well, you could get the reactive oxygen species. Now, mostly when reactive oxygen species cause trouble, they cause trouble by inappropriate signaling, and that can lead to perhaps aging, accelerated aging, vascular dysfunction. In their most extreme case, they can directly damage the cell and cause cell death. That often happens in reperfusion injury following a heart attack or a stroke. So what can we do about these? Well, we can deliver antioxidants. Now, there are two types I'll talk about here. MitoQ, which is related to the CoQ talked about earlier, but is a mitochondria-targeted version, that can mop up the reactive oxygen species. They're being formed, but they're being mopped up. And so it behaves as though they're only being formed in the right amount and everything returns to normal. And the second one, dimethylmalonate. That stops the formation of reactive oxygen species under certain conditions. So those are two ways in which we can intervene. Now let's focus on delivery of compounds to mitochondria. Now, mitochondria are complex organelles. It's important to deliver to the right place. You don't want to deliver to the intermembrane space if what you want to get is the things to do with the DNA, the carboxylic acid cycle, fatty acid oxidation, etc. What you want to do is deliver to the mitochondrial matrix. And my favorite way of doing this is to use the lipophilic cations, in particular triphenylphosphonium, which not only because it's lipophilic allows it to cross both membranes, but because it's positively charged, it accumulates over 1,000-fold in the right place. And that means you can deliver your drug to the right place for the right action. There are some restrictions. You can ask me about those in the chat. Thanks very much.
Thank you very much, Richard. We move on now to Stefan Jakobs. Already we discussed a lot about structure of mitochondria, and Stefan is going to present some beautiful work.
Yeah, hello. I'm Stefan Jakobs from Göttingen. Here on the left-hand side, we do see an electron microscopic image recorded with classical electron microscopy. We do see the infoldings of the inner membrane, the so-called crystal. And we also see, maybe a little bit worse, the outer membrane which surrounds this organelle. Well, these images have been recorded already 50 years ago, and they gave rise to the cartoons we now know or now see in every biological textbook. We do see the crystal, the outer membrane, and here is the DNA packaged internucleoid, which is found within the interior of the mitochondria between the crystal. Well, at least for me, these images or these cartoons of mitochondria somehow look like a dull organelle, relatively simple. Well, this is very wrong. And in the next few minutes, I would like to show you images recorded with modern instruments that show how dynamic and how complex these organelles are in reality. Let's start with modern electron microscopy. Here we do see a reconstruction of data recorded by a method called FIPSEM electron microscopy. It's a method that allows to record images in 3D. So we do see the outer membrane in transparent, and in the interior in blue here, we do see the individual crystals, the invaginations. One can acknowledge that they are nicely stacked, although the individual crystals have quite a bit of heterogeneity. Electron microscopy is very strong in imaging membranes. However, if you want to see individual proteins or living mitochondria, then we have to switch to light microscopy. This here is a typical image of a light microscope of a human cell decorated with antibodies so that we can see the antibody, the nucleus in blue. We can see the cytoskeleton in gray and in green. And this yellow-red structure here, that are indeed the mitochondria. The mitochondria are not, at least not in general, act like organelles, but they are tubules and they can form networks. Well, here's another light microscopy image. And in fact, there was a challenge for all microscopists for the last 100 years or so, because there was the concept that it would be impossible to visualize structures which are inside of the mitochondria. Well, science changed this view. By now, we have methods that allow us to visualize structures within the mitochondria, and I would like to show you the proof. So this image here is the conventional microscopy, and with super resolution microscopy, we already can see here in this overview much more detail. And when we zoom in a bit here, and then we zoom in even more, then we can tell that we have been labeling these mitochondria here for two different proteins in red and in magenta. And you may acknowledge that they have different distributions. Still, especially in this region here, they seem to form a striated pattern. And this indicates us how they work. They seem to be a part of what we believe is a skeleton in these organelles. However, this is a fixed cell. What is about live cells? Well, we also can record with modern microscopy living cells. So here we do see parts of a cell. We do see the mitochondria in this human cell. In gray, we do see the cristae. In interspace between the groups of cristae are individual nucleoids here in red. So what I think, what we can learn from these modern microscopy techniques is that mitochondria are dynamic, they are complex, and they are very beautiful, at least in my eyes. But why are we studying that with these modern microscopies? Well, it is very well known that architecture and function are closely linked. So we are very sure that by these new imaging methods, which allow us to look deeper into the structure and dynamics of these mitochondria, we will be able to get a deeper understanding of the function of mitochondria in health and disease. And if you have more questions to this, I would be more than happy to answer that. Thank you.
Thank you very much, Stefan. Obviously, now that leads us to the next question and the next speaker, the interactions between all these proteins and how they keep their structure within mitochondria. And Hana Antonicka from McGill University is going to talk to us about omics approaches, so data science approaches to actually define mitochondria protein interactions. Hana, over to you now.
So I will thank you, Kostas, and the FRSE Society for organizing this virtual meeting. And I will talk about mitochondria in the age of the big data. So as we have heard, mitochondria are very complex organelles, and identifying a mutation in a gene causing mitochondrial disease is only the first step in a long process of understanding what are the consequences of the mutation in the cell. And in recent years, the advancement of several scientific techniques allowed us scientists new opportunities to study the effect of mutations. We can now create cells which would carry a pathogenic variant and compare them to the wild type cells in order to collect data on the changes which happen in adaptation to the presence of the pathogenic variant. And we can look at the changes in the expression of the genes by transcriptomics, in the changes in the levels of proteins by proteomics. We can look how these small molecules change, ATP, lactate, coenzyme Q by metabolomics, and how do the lipids change by lipidomics. So the suffix omics means that all constituents are considered collectively. And often the gathered data sets contain thousands of genes or hundreds of proteins or metabolites. So how do we interpret this data? We use statistical analysis, where we can integrate all omics to try to understand the relationship between them. And as part of my research, we try to understand the functional relationship between mitochondrial proteins by determining the interactions between them. So as mitochondria contain more than 1,100 proteins, and as we heard by Bob, about 300 of them have been linked to mitochondrial disease. These proteins perform their function within their mitochondrial compartment, but they do this by interacting with their functional partners within a pathway or as a component of a complex. And they are also, though, part of the environment because they interact with their neighbors. And to identify these interactions, we used a technique called BioID, or proximity biotinylation assay, in which the protein of interest, in our case a mitochondrial protein, is fused to an enzyme. And after expression of this fusion protein, the enzyme can attach a molecule of biotin to its proximity partners, the neighbors. And it would be these two proteins, but not to a protein which is further away. Biotinylated proteins, they can be then captured and identified. And when we perform this technique using 100 selected mitochondrial proteins to create a mitochondrial protein proximity map. And the data which we collected, the analysis enabled us to create networks. This kind of webs, like an example is shown here. And now we can map individual proteins to these networks and to their subcompartments within the mitochondrion. And we can also identify novel mitochondrial proteins. We were also able to assign proteins to individual functional networks, which will help us to understand the consequences if mutations in genes coding for any of these proteins occur. What we also detected were interactions of mitochondrial proteins with other organelles in the cells. And this is very important for the communication in the cell, as we heard already from Richard about. So with this framework set, we are now interested in determining how is the mitochondrial proximity network rewired in the presence of a pathogenic variant. So we are going back to looking in the pathogenesis of the disease and to try to understand further and maybe in the future to provide targets for future treatment strategies. Thank you, and back to you, Kostas.
Thank you very much, Hana. Beautiful presentation. The time we have lost 2 or 3 minutes, we're going to take from the break, so it's not a problem. So now we move on to Bruno Chausse, who is actually in Heidelberg, but he comes originally from Brazil. He's going to tell us a little bit about his current research now in metabolism, immunity, and the brain, the human brain, and also give some perspective as an early career researcher in transitioning now from postdoc to independent position. Bruno, to you.
Hello, everybody. Let's start the talk today. I'll basically talk a little bit about my experience as an early career researcher with mitochondria and my scientific development. And later, I will talk a little bit about the new things we are discovering about mitochondrial biology and physiology. Basically, as you can see in the left side of my slide, this is quite a small story about my pathway in science. And we always have to deal with a lot of moves, a lot of challenges to build up our scientific identity that basically is will be the support of our next step in science, have our own lab, our own research group. And it's interesting that our knowledge about mitochondria grows following up this development in the pathway also. And as you can see, we start to see some basic aspects of mitochondria at the beginning of the career. And we are able to increase the complexity of our analysis by experimental setups and later by complex inferences about mitochondrial function and how mitochondria can change the function of our cells, of the organs of our body. And basically, with this knowledge, we are now ready with this substrate, with this support. We are now ready to start our new career, our new moment as a principal investigator with our own lab. And so how about you, Bruno? What you did in the past? What is the way you are looking for? So basically, in the last years, I was like accumulating experiences experimentally and theoretically to work with physiology. Specific brain physiology, how alterations in specific aspects of our brains change the brain function and health and diseases. It's important to highlight today that the brain is not composed only by the nerve cells, so by the neurons that usually are the most studied and famous cells. We have a lot of different cells that are really important to keep the brain function going on. So we have microglia, they are the sentinels, they are the guards of the brain that are activated when the brain is challenged. We have astrocytes that support the brain, giving molecules and metabolites to keep the brain function. And we have the neurons. They are the effectors. They are the hard workers of the brain. And interestingly, very simple alterations in microglial biology and in mitochondria specifically can completely change the relationship between these cells in the brain. So I will talk today about the metabolic reprogramming of mitochondria, specifically in microglia, and how it changed the function of these cells and how it changed the coupling between the cells in the brain. Basically, in mitochondria, we have a lot of metabolic pathways that you can see like streets where the metabolites, the cars of the metabolic pathways, runs through and performing transformations that will support the mitochondria and the cell function. Interestingly, when we have damage or challenges in the brain, microglia identify these challenges and they are activated. And the interesting thing we saw recently is that this new profile of microglia is supported by alterations in mitochondrial metabolism. So the older streets inside mitochondria, they are modified, they are interrupted, and they create specific new pathways that support this new state, this new phenotype, this new state of the microglia. And it's really interesting that according to these interruptions in the streets, so in the metabolic pathways inside mitochondria, we can have a protective phenotype or a damaging phenotype of microglia. So according to specific alterations in mitochondria, we can have protective or neurotoxic microglia. And basically, these are the new things we saw in the last years. If you want to know more, we can go for the reference here. You can also follow us in Twitter or send email if you have more doubts or questions about the system. Thank you very much for the opportunity. See you soon.
Thank you very much, Bruno. And that concludes our first session with the speakers. That I would like to thank all of you for trying to simplify very, very complex issues in 5 minutes. So before going to the Q&A, I would like to make just a very brief couple of points. First of all, we don't have the time to answer all the questions that are coming through the Q&A. We'll try to group them together so that we can answer as much as possible. And also, due to the nature of the event, we cannot comment on personal cases. I would like to start off the discussion by asking a very generic question to all the speakers, and then we will move on to individual questions by the audience. So we heard a lot about mitochondrial research. Where do you think we go from here? So where do you think are the future challenges in the field? If any one of the speakers of the first session would like to talk, you can just switch on your microphone and have your comments on that.
So, Kostas,
yes, Bob. Yes, please go ahead.
I guess I'm a little bit I'm a little bit biased, but I really think that the mitochondrial field has made such wonderful progress over the last few years, and the one area that we're really sticking on at the moment is curative therapeutics, and I think this is a huge question, and I really think that the way things are shaping up at the moment, that we should be looking at some very interesting possible therapeutics in the next few years.
Thank you very much, Bob. Anybody else to comment on this question?
In terms of
Richard?
In terms of secondary mitochondrial diseases in mitochondrial dysfunction, getting tissue-specific or organ-specific hitting of the problem is going to be key. So specific mitochondria targeting, but within a particular organ.
I completely agree with that. I also think in terms of the targeted that you mentioned, I think we need ways to actually target within mitochondria with very, very specific resolution to get the probes that we can make to get the activity we want at exactly sub-mitochondrial localizations. Anybody else? And then we'll move on to the Q&A. No? Okay, so for the Q&A questions, we have a couple of them. My colleague, Dr. Erica Fernandez-Vidara from my lab, a research scientist in my lab, will help me trace all these. So Erica, if you can just go with the first question from the Q&A.
Hello, everybody. So the first question is for Bob Lightowlers, and it's from Hirosaku Arimoto, and he would like to know that being that mitochondrial DNA has heteroplasmy and that dysfunctional mitochondria are selectively excluded by selective fission, are mutations in mitochondrial DNA selectively separated with the damaged mitochondria by these mechanisms?
So thank you very much for the question. It's a very interesting area, and it's certainly an area that several people are approaching, and also are approaching in different animal models as well. So the question is whether or not we could perhaps speed up mitophagy, which is the system that you're discussing that is potentially removing the dysfunctional mitochondria. And there is a whole great big area of interest now in identifying compounds that can promote mitophagy, and to see that to address that very question as to whether or not you can modulate mitophagy in this way by modulating heteroplasmy.
Thank you, Bob. Yeah, we can move on to the next questions.
That's another question for Bob, now that he was speaking. Joanne Kotelawala would like to know if there's a way to identify pathogenicity of the mitochondrial DNA control region of variants, for example, in silico analysis.
Hi, thanks very much for the question. Yes, I mean, I'm not the world's leading geneticist or genomicist, but certainly there's a lot of interest in trying to identify point mutations, substitutions, which can affect or partially modulate the effect of known pathogenic mutations in the mitochondrial genome. And in fact, it's been an area that people have been interested in for many years. I'm not entirely sure that there are, as yet, really well accepted pathogenic mutations or substitutions in the control regions that really do have a modulation on disease. But certainly people are very interested in that area.
Thank you, Bob. I think there are a few questions for Stefan Jakobs.
Yeah, there's one question about how do you visualize nucleo, or how many nucleodes do you typically see in a single mitochondrion?
Oh, that's indeed an interesting question. I can answer how many nucleodes I see in a cell. That are between a couple of hundred to a few thousand. That depends really on the cell type, whether it's, for example, muscle or a neuron. The number of nucleodes per mitochondrion is more difficult to answer because mitochondria can fuse and divide. So at any stage that might differ. There might even be mitochondria without a nucleoid, at least for a while. So because they're so dynamic, that is very, very different and will be different under different physiological conditions.
Thank you, Stefan. There's
But
also...
there was a second question. It was how can you maybe specifically stain cristae to visualize them by light microscopy?
Yeah, that is something we as a field can now for maybe 2 years or so. There are two possibilities. One is dyes that specifically label the inner membrane of mitochondria. Mitochondria, the inner membrane is rich of cardiolipin. It has a specific composition. So we have dyes that go specifically into the inner membrane and thereby also into the cristae, and we can visualize them. Or we could use proteins which we genetically tag so that they're integrated in the inner membrane, and then we can label this tag. So both methods work, and we can visualize them together, but only with super resolution microscopy. Otherwise, we wouldn't be able to see them and to resolve them.
A couple of questions for Hana and her approach in terms of the bio-ID and interactomics in mitochondria.
Agna Singh would like to know how can you compare the disruption of protein networks between the healthy and the disease variants?
So we can profile the interactors, the proximity interactors of the pathogenic variant, and then compare it to the wild type. I saw that there was a question about what the wild type is. So that's the variant which is not mutated. So it's the common variant present in the cell. And when we get the data, we get the interactors, we also can quantitate what the interaction is based on the data which we receive. And then we really compare them to each other. And usually what we see, we haven't done big sets yet, but we can see some new interactions which are only present when the pathogenic variant is in the cell, or we can also see what interactions are lost. So the interactions which is only present with the wild type, so the normal variant, is not present in the pathogen, and that's how we compare them.
Also, Hilario Ruelas would like to know what kind of different data you can obtain by bio-ID compared with other physical interaction approaches. So
the other physical interaction approaches, what probably she or he is referring to, is really pull-down experiments or immunoprecipitation, as we call them. And in these cases, we usually just pick up very strong interactions, and because we have to use chemicals to disrupt the cell, the mitochondrion, to be able to pull down the protein of interest, we often also can disrupt the interactions. The bio-ID experiments are in live cells. The labeling happens then and only after the cell is disrupted. So we can often detect interactions which are just very short-lived, or interactions and proteins which are not that abundant because they will all get labeled within the cell.
So there is, thank you very much, Hana. There is an interesting question for everybody, which was placed by Julia Garcia, saying, how much do you think mitochondrial therapies can be applied to chronic disorders where mitochondrial dysfunction is observed, but is not necessarily the central aspect of the disease?
So I think they can be applied very well. What's really good about the mitochondria is being the central hub of metabolism. A lot of pathologies are feeding their problems through mitochondria. So if you can get the mitochondria to behave well, you can stop the pathology, even though it's happening, from having its effect. Now, there has been a clinical trial on MitoQ and vascular function in healthy older adults, improving their vascular function. And I think that's effectively how that's working.
Yeah, can I just follow up from Richard? I think that that mitochondrial disease is a very good surrogate, if you like, for dysfunction, chronic dysfunction in many other neurological diseases. And I think what's really what could be really potentially exciting is that you can run nice, discrete clinical trials with mitochondrial disease patients, as long as you've got enough patients to be able to interpret your results. But I think it's a very good surrogate.
In terms of there is also another question coming in the Q&A that is a little bit related to that. I mean, how can you target, once you know how to target a specific cell or tissue, how can you target from a mixed population of healthy and unhealthy mitochondria, how can you target the unhealthy ones as opposed to the healthy ones?
So so let's just look at I'll give you just a general answer to that. Look at the evolutionary background of mitochondria and think of these as separate, bacteria-like things competing against each other. All you have to do is be able to give, within the heteroplasmic thing, a selective advantage to one set. So you look for the difference and you aim for the difference to make it selectively disadvantageous for its reproduction.
Yeah, and can I just add a little bit further again with Richard's point? As Stefan's been talking about and just showing this very, very beautifully, this idea of mitochondria have discrete organelles in many tissues is not the case. I mean, there's massive amounts of fission and fusion and networking. So it's quite tricky to actually even think about, conceptualize mitochondria or discrete mitochondria unless we think about them budding off because they're dysfunctional and they're about to be eaten by mitophagy.
Okay, there are one more question, Erika.
There's a question by Mustafa that he says it's a naive question, but I think it could speed up it could trigger a little bit of discussion. Like if you think that there are some mitochondrial mutations that can confer positive outcome, so if can specific mutations confer through evolution history benefit for individuals having them? Anybody?
Maybe I can just put in my sixpenneth, but I guess we have been doing a very large and extensive evolutionary experiment, and what we've done is selected potentially for those positive mutations, if you like. And I doubt whether we're going to be able to get anything too much better.
Okay, we have one more. We have time for maybe 2 more questions to discuss. There's a specific one for Richard about ROS markers. What is the best marker for ROS to assess damage in mitochondria?
It depends what you mean. Are you assessing the damage? Now, so my area of expertise would be looking at the actual ROS production, and I would be using small sensors like a probe like Mito-NeoD or Mito-B, which can be used on whole organisms to look at the mitochondrial specific hydrogen peroxide and superoxide. Other people will probably have better markers than me in terms of protein, so I'll pass it over to see if anybody else has something better.
Okay, I think we will take one final question, Erika.
One for Gloria. in terms of what is the efficacy and the maximum amount of coenzyme Q that can be given to patients that have primary coenzyme Q disorders?
Well, it depends on the weight of the patient, but in clinical trials, in a big clinical trial, has been tested up to 1,200 milligrams per day. That it can be even higher, and but not all the patients respond positively anyway. It's very hard the coenzyme Q to get to the proper place and work actually, because of the lipidic nature of the molecule.
Okay. Good, excellent. I think there are a lot more questions in the Q&A, but I think we need to stick to the time because we have also the other session coming up. So now we will take a break of 10 minutes, and then we will resume with the second session, which will be on societal issues and impact on the society of mitochondrial disease and mitochondrial research. So I would like to thank at this point all the speakers of this first session. It's very difficult to stick to 5 minutes when you want to explain, all the intricacies of the research that is very, very complex. So I want to thank you for that. I want to thank the audience, the attendees, for the very nice, insightful questions. And we'll resume in 2:10. Thank you very much.
Hello, and welcome back to part 2 of this Royal Society mitochondrial event. I'm Simon Woods, I'm Professor of Bioethics at Newcastle University, and it's my pleasure to chair and present in this second session, in which we're going to explore some of the wider societal issues associated with mitochondria and mitochondrial disease. We'll follow the same format with several speakers giving short presentations, and there'll be an opportunity for you to ask questions in the Q&A, and we'll deal with those at the end of the discussion. So without further ado, I'd like to introduce our first speaker, Dr. Lyndsey Butterworth, who's Head of Science Communication at the Lilly Foundation, and she's going to talk about the patient perspective.
Great. So as Simon said, my name is Lyndsey, and I work in a science communication role with the UK's leading mitochondrial disease charity, the Lilly Foundation. So the charity itself was founded by Liz Curtis back in 2007 after she tragically lost her daughter Lilly to a mitochondrial disease. So at that time, there was very little information available about mitochondrial disease, and certainly very little in the way of support for families with affected children. And so that was when Liz and her family set about changing this, and the Lilly Foundation was founded. So in the beginning, there was just that one family, but the charity has grown significantly over the years, and we now support nearly 1,000 patients and families from across the UK who are affected by mitochondrial disease. So our mission at the Lilly Foundation is to improve the lives of those affected by the condition, and we actively pursue this in 3 key ways. So firstly, we provide essential support to patients, families and carers. Secondly, we educate and inform and raise awareness of mitochondrial disease. And thirdly, we fund scientific research. And we really want to do this to advance the search for effective treatments and ultimately a cure for mitochondrial disease, which is really kind of the main goal of our charity. So we are very proud that to date we've invested over £2 million into mitochondrial research. And this is not only to improve the diagnosis of mitochondrial disease, but also to fund research into potential treatments. And some of these projects are being co-funded with both national and international patient organisations. And I believe Elia is going to speak to us a bit later about the importance of these collaborations. So let me see if I can share my next slide. Great. So So I was asked by Kostas to talk a little bit about the patient perspective and the importance of engaging patient groups with all aspects of mitochondrial research. And this is important as it can provide researchers with a different perspective. It can help improve their research or help redefine their research priorities and ensure that their research has that real patient focus. And I would like to mention briefly about a partnership we've recently been involved in with Genetic Alliance UK, which was a priority settings partnership for mitochondrial disease. And this was a really extensive process that involved lots of different patient organisations and medical professionals who helped identify the top 10 research priorities for mitochondrial disease. And this is available in a fantastic final report. And I would really encourage people or researchers who are looking to apply for funding or who are wanting support for grant applications to access that and have a look. And we can provide the link after these talks. So at the Lilly Foundation, we work very closely with researchers from all around the world, and we have many different opportunities for researchers to engage with our patient community. And one example of this is our Lilly Family Weekend, and this is an event that we hold every year, and it's a really unique opportunity for families to come together with the doctors and the researchers to really learn more about mitochondrial disease in a very informal and relaxed setting. And one of the most popular parts of this weekend is a family fun weekend, and we encourage researchers to come along and engage the families with their research by hosting researcher stands. So this is kind of fun and interactive games that can help get across sometimes quite the complex aspects of mitochondrial disease. But like I say, this is something that the families and the patients really enjoy. The researchers get a lot from it as well, and it really is a fantastic atmosphere and a wonderful kind of all-inclusive feeling when everybody's together. And we also host patient engagement days with a number of the highly specialised mitochondrial centres across the UK. And again, that is another wonderful opportunity for patients to come together with the researchers and the medical professionals to engage with some of the research that's being done. And again, those researcher stands are very popular. So they're often contacted by researchers who tell us about their research plans, and they ask us for our research priorities as a patient organisation, and we are given opportunities to provide input into the study design. And then we are also able to share that information with our patient groups, both through our website and through our social media. And this can be very important if you have a research project that needs some participation. So if you need people to sign up to help with research projects or trials, that information can be shared within our patient group. And that's a really fantastic resource. And again, we'd have a page on our website, and I can share the link to that where we can post that information. So again, if there are any researchers who need patient participation, we'd really encourage you to either get in touch with myself or with Alison Maguire, who is our head of research at the Lilly Foundation. So this, I guess, kind of two-way dialogue is really important, not only for the researchers and for the research itself, but it also really empowers our patient community and provides them with an opportunity to share their experiences and to have their voices heard. Like I've explained, this can be important for research, but it can also be important for different things such as policy issues. And I just one of our greatest achievements at the Lilly Foundation, which was changing the law to allow mitochondrial donation in the UK. This is a novel IVF treatment, and like I say, it was really the patient voice and the patient experience that was crucial to the debates and the discussions that took place around this technique. And the law was changed back in 2015 to make it available to some women who are at risk of transmitting a mitochondrial disease to their children. And like I say, this was a real it was the patient voice that really kind of gathered a lot of support for that. And we have a wonderful video that, again, I can share the link to that just to highlight kind of our involvement in that and how we could support patients to take part in those discussions. So with that, I would just like to pass back to Simon.
Thank you, Lyndsey.
You're welcome.
And I would just like to then introduce our second speaker, Elia van der Veer, who is Chair and President of the International Mito Patients Organization, and she's going to talk to us about the international patient perspective.
Yes. Hello, everybody. I'm going to talk a little bit about international Mito patients. The Lilly Foundation is a member of IMP, and IMP is an international network or federation, if you will, of national patient organizations, all involved in mitochondrial disease. IMP involves only patient organizations and nothing more. Because the disease is rare and represents relatively small groups in each country, it made sense to organize ourselves on an international level. And it turns out to be so important. Our mission is to improve the quality of life for mitochondrial patients everywhere in the world. The ultimate improvement of the quality of life, of course, is a cure, but until then, also much attention is required for the best of care for patients and for smoothing the path towards a cure. So what do? We collaborate cross-border, we share information, we share best practices with all the stakeholders in the mitochondrial community, being clinicians, researchers, other professionals, the regulatory authorities, industry, and of course, patients and their families. Here you see all our member organizations, 16 in total. As you can see, many of them come from Europe, but we also have member organizations in Australia, Canada, and the United States. The latest members are from Finland and Bulgaria. IMP has contacts with many more countries in the world. Patients and families really know how to find us. And we know that because there's patients from more than 35 countries in the world participating in an IMP closed Facebook group. With our members in total, we represent roughly 10,000 patients in the world. And as a result, we can be a powerful partner and give the patient a louder voice. It is a bit black and white, but you can say that researchers and clinicians know everything about the disease, and we know everything about the burden of the disease in everyday life of patients. And as such, I really believe we are natural partners. Some of the activities we do successfully are mentioned here. We have a very good service to support individual patients from countries where nothing is being organized for them. Think of a patient on a farm in Argentina or in a small village in Russia or a big city in Africa. All of them find their way to IMP and ask for help. We participate, initiate, and facilitate scientific workshops. The last one being a scientific workshop on the development of a list of medicines to be used with care, and that one is published on our website. We are often asked to endorse grant applications for research. We sometimes participate in studies as a partner and sometimes initiate a study ourselves, such as a German study into chronic pain with adult mitochondrial patients. And last but not least, we initiated the World Mitochondrial Disease Week, which is this week. Most of the activities are taking place on national level, organized by the national patient groups, but there are some global activities as well, such as the Light Up for Mito campaign. Thank you for listening. Back to Simon.
Thank you, Elia. And for our next speaker, it's a familiar face from the science panel, but speaking in a very different context. I'd like to reintroduce Dr. Gloria Brea Calvo, who's going to speak about the Mito Women Research Network.
Well, it's a pleasure for me to be here to present now the Mito Women movement, the Mito Women initiative that was just set up last June. You are probably aware of the decreased presence of women along the academic path. As you can see here in the famous gender scissor chart from the Chief Figures report from the European Commission. The situation is perfectly represented by the leaky pipeline cartoon here in your right. So women face lots of barriers to be retained in the academia, and also to get to leadership positions. And the factors contributing to this situation are many, and some of them, for example, are the unconscious bias, the lack of visibility, also lack of role models. And this leads to the well-documented situation by which women get fewer citations, that they are less, they get less invitations as keynote speakers to some conferences, although this is changing slowly. But in general, all this is provoking the general lack of motivation to some of them, and difficulties in the career promotion. So we wanted to make a change, and we decided to create the Mito Women initiative. It's a self-organized movement, and we firstly created a web page where you can see our searchable database with all the community, where the community is represented. You can search, you can look for women working in mitochondria by the name, the position, the university, and also keywords. So this is a great tool for everyone who needs to get in contact with an expert working in different fields of the mitochondria science. Another, we are very proud that we are now almost 300 members in the community and growing. So if you want to join, you just have to go to the web page and go to the form to join the community. The other great tool that we have is the Mito Women map. So you can look where all the women working in mitochondria are around the world. As you can see here, of course, we have, unfortunately, a very big gap to close. And this is actually one of our main goals, actually is our priority now, to close this gap, which reflects actually the lack of connections that some parts of the world have with others. And this is something that we want also to work to fix that actually a little bit. We also have a Twitter account, the Mito Woman, you can follow if you want. And recently we created a workspace in Slack where all the community share opportunities, advice, do some networking, and yes, we with this tool we look for an improvement and to boost the networking and the mentoring among the community. So with our initiative, this Mito Women initiative, what we want is to promote equality, visibility, and the networking of women working in mitochondria. So we contribute for them to not leave academia as much as we can. And thank you very much. I hope you find it interesting. Thank you.
Thank you, Gloria. And now I'd like to introduce our next speaker, Dr. Kathy Herbrand, who's reader in medical sociology at De Montfort University. And Kathy is going to talk to us about sociological perspectives on mitochondrial disease.
And I would like to address here some of the challenges faced by families affected by mitochondrial disorders when it comes to deciding to have children. In a context often characterized by a high level of uncertainty, what are the factors influencing women and couples at risk of transmitting the disorder? To examine this question, I drew on the in-depth interviews I've conducted with families affected by the mitochondrial disorder that were either maternally inherited or due to nuclear defects. As you know, mitochondrial disorders raised particular attention during the UK debates on mitochondrial donation, since this new reproductive technology was often presented as the solution to help any women at risk of transmitting the disorder to have a healthy child. However, the accounts I collected during my research revealed a much more nuanced and complex picture, where decisions were not straightforward, but were guided by particular ideals and depended on various constraints. I just would like to mention here three key elements which could challenge women's reproductive decisions and tend to be overlooked or ignored in public debates and medical accounts. The first difficulty met by family affected by mitochondrial disorders was that of accessing and understanding the complex genetic and medical information surrounding the causes of the disorders, the transmission risk, and the implication for the reproductive options and for the future offspring health condition. The parents I interviewed often complained about the difficulties of assimilating a large amount of information during genetic counseling, and often ended up confused or conflicted when they were either told that it was not possible to determine the risk of transmission, or when they had to try making sense of specific risks, such as 1 in 4, which could be interpreted variably. In many cases, it was also difficult to predict the extent to which the offspring might be affected. The medical information that future parents receive could therefore generate more of a sense of anxiety than a sense of relief and solutions. Moreover, searching for advice online often made things worse. Secondly, it's important to consider prospective parents' experiential knowledge of the disease. This means the knowledge and perception of the disease gained either through their direct personal experience or through that of a relative affected by the disorder, often their child or their mother. Participants were often aware that mitochondrial disorders affect each person differently, even with the same mutation and within the same family. However, experiential knowledge was key to assessing what they felt they could accept and deal with. For example, taking care of a child in wheelchair, but he also informed what they thought they were not able to deal with, such as taking the risk for the existing children to be traumatized by the prospect of losing another sibling. It is important to note that being affected by the disorder or having lost a child from it did not necessarily mean that patients abandoned the idea of having children. Often, it was precisely what was giving them hope and purpose, or would enable them to reinstate a sense of normality in their life. A third challenge faced by potential parents was the temporary or permanent impossibility of accessing reproductive technologies that would prevent the transmission of the disorder. This could generate a great amount of frustration among women and couples who wanted to have a biological child, which was the case of most of my participants. This situation often happened in relation to mitochondrial donation, as this new technique remains inaccessible to most women at risk for various medical reasons. But pre-implantation genetic diagnosis and prenatal diagnosis could also be inaccessible because of the couple-specific mutation had not been identified yet. In addition, financial constraints could limit access to the technology. For instance, if a couple already had a healthy child, they were not entitled to NHS funding for PGD, and often they just could not afford to pay for it. So these are only a few of the challenges faced by families affected by mitochondrial disorders. However, I hope this brief presentation helped highlight the complexity of the decision faced by these families and the need for these various social, psychological, and financial constraints to be considered further in policies and clinical practice. Thank you.
Thank you, Kathy.
Yes. Okay, if Gemma and Sneha can start the presentation.
So in the first session, we heard from the perspectives of people, experts in science, and as highlighted in some of the presentations here on patient engagement and voice, clearly people living with mitochondrial diseases, their families, and the health and care professionals also have invaluable support, invaluable insights to offer. At GSA, we start from the perspective of the people who have a lived experience of a condition. Using ethnographic methods, we understand what it's like to live with a disease, what helps them, and what are the challenges that are most important to them. Their perspectives are vitally important to help set the agenda for the research and the innovation. To date, we've not focused conversation specifically on the topic of mitochondrial diseases, but we have worked with people with living with long-term conditions, such as diabetes, and hopefully you can see the relevance of design in wider mitochondrial disease contexts here. And as designers, our focus is to identify opportunities for new ways of supporting people living with a condition, which can lead to impacts on the quality of life and the quality of future care experiences.
So how do we do this? Many of you would have heard the term co-design, which is becoming a bit of a buzzword in the public sector. But what this means in practice can significantly vary. At the Glasgow School of Art, by co-design, we mean fully opening up the design process and meaningfully involving people who will be impacted by an innovation in its conception and development. This means starting from people's lived experience and working together with multidisciplinary experts to identify opportunities and generate new ideas. This can mean bringing together people with disparate or conflicting perspectives. And the role of design is in enabling agonistic but ultimately productive dialogue. As designers, our skills are in visualizing these different perspectives and helping to reframe the challenge, translating through thorough understandings of the context into opportunities for innovation. We can also use these understandings and visual skills to make new scientific developments meaningful to people, enabling them to see how it might impact on their lives.
So in our experience, people can often tell you about their challenges, but it's very hard to imagine how things could be different. So the role of the designer is in listening and translating insight into opportunities and ideas and making these tangible so that people can be involved in their development. Future experiences are brought to life through prototyping and made relatable through future scenarios. This enables critical reflection on what is of value and how it can be made better, enabling the development of person-centered and usable innovations.
Within our work in health and care, we focus on person-centered innovations that can enable, for example, self-management of a long-term condition, high-quality interactions between the person and their health and care professionals, multidisciplinary working between health and care professionals, and support to navigate the complexities of the health and care system. So we hope we've whet your appetite to learn a bit more about the role of design in health and care. You can find out a bit more about what we do on our project repository, which is futurehealthandwellbeing.org. And I'll hand back to Simon. Thank you.
Thank you.
So in 2020 is the centenary of the birth of Henrietta Lacks, an African-American woman who died of cancer in 1951. Her name's well known following the book and film about her life, but for many years, her cells were better known than her story. HeLa cells are one of the first human cell lines to be successfully grown in a lab and have contributed to research of major benefit to human health, including research into mitochondrial diseases. An editorial in the journal Nature points to some of the ethical complexities behind Henrietta's story. Her tissues were taken without consent, commercially exploited without compensation, or even acknowledgement to her family. The editorial picks up on the themes of Black Lives Matter and points to her story as an example of exploitation and injustice. It calls for a greater awareness of the ethical complexities of medical science. Since the 1950s, there are many international regulations and laws setting high ethical standards for the use of human tissue in research. Informed consent, opting out, disclosure of findings are all common issues. The Wellcome Trust Centre for Mitochondrial Research at my own university in Newcastle has been pioneering research into treatment for mitochondrial diseases with scrupulous attention to ethical standards. Things have certainly progressed from the 1950s when patient consent was not considered relevant, when ethical review of medical science was not a requirement, and when medical research was not considered appropriate for open and public debate. Now I turn to mitochondrial donation and some of the ethical challenges of this. One strategy for reducing the chances of mothers transmitting mitochondrial disease involves intervention in the process of family making. Although IVF is a standard treatment for infertility, its adaptation to avoiding the transmission of faulty mitochondrial genes is recognized as challenging, not only for technical but for social and ethical reasons. Put simply, a fertilized egg from an unaffected woman, a donor, has the nuclear material replaced with that of an affected woman, the recipient. The fertilized egg containing the healthy mitochondria is implanted into the affected woman to avoid the vertical transmission of mitochondrial disorders to her child. So following this, I'm going to talk about three ethical issues that arise from this kind of technology. The first is family making. One important benefit of these techniques is that mitochondrial donation will allow parents to be both genetic as well as social parents for their child. However, critics argue that genetic parenthood is less important than social parenthood, and there are other less complex and controversial ways of becoming parents of a healthy child. Terms such as three-parent embryos, three-parent babies have been used to capture these concerns. But supporters of mitochondrial donation downplay the importance of mitochondrial genes because they are such a small proportion of genetic material. They're not associated with the complex traits that make up a person that are coded by the nuclear DNA. Others argue this is a misleading simplification. 2, germline modification. This technique introduces heritable changes, especially where the resulting child is a girl. Mitochondrial DNA is passed down the female line and may permanently change the family's genetics in the future. Germline modification has always been a red line to those with concerns about playing God with the human genome or concerns about eugenics. Supporters argue this can be managed by careful regulation for therapeutic use. Those who are cautious point to the uncertainty of risk compared to the well-established and low-risk alternatives. Others are concerned that once the red line is crossed, germline modification will become routinely used, perhaps even for more problematic interventions, such as human enhancement. And 3, recognition injustice. Recognition injustice occurs when individuals are not given the full recognition they deserve. This echoes one of the concerns about Henrietta Lacks, that she was never really acknowledged. Similar concerns have been raised in the context of mitochondrial donation, pointing to the hidden labor of the women who provide eggs for mitochondrial donation at considerable inconvenience, discomfort, and risk. The seemingly neutral language of bioscience can also conceal moral complexity. Human tissues have been of great value to science and even of great commercial value to scientists, but have been described in terms of low value as waste, spare, or even surplus tissues. And a similar issue arises in the public information approach to mitochondrial donation. Playing on a popular analogy of the mitochondria as the power source of the cells has the potential to reduce the significance of the donated mitochondria to that of a replaceable battery. In comparing mitochondrial donation to a replaceable battery, the women donors may not be afforded the same status as other egg donors. They're rendered invisible. We all have an interest in the future conduct of potentially beneficial and pioneering research, but we have a stronger interest in the open, honest, and ethical conduct of science, open to public engagement and critical scrutiny. Thank you.
Okay, thank you very much, Simon. So if you can now take on for handling the Q&A question for this session.
I think I'm going to be joined by Michael, is that right?
Yes. How do we do?
Hi.
So I'm also from the Innovation School, so part of the co-organisers with Kostas. So happy to thank everyone for their presentations this afternoon. Really, really diverse and interesting perspectives brought together there. Not sure if there were questions that came in from the Q&A. There was one I saw for yourself, Lyndsey, about the Lilly Foundation, and I know it's UK based, but what other countries has its work kind of developed with as well?
So I guess just kind of as Elia mentioned, we're part of the International Mito Patients, so we work with a lot of different patient organisations from right across the world. And I guess that's important with any kind of rare disease. It's really a kind of way to come together and make sure kind of patient voices are heard and things. Like I mentioned, we co-fund some projects, so we work very closely with the Mito Foundation in Australia, and we co-fund some research projects with them. But like I say, yeah, it's international, and we really do all pull together through IMP to work together.
Excellent stuff. And can I actually ask, because it's a perspective we haven't quite touched on in detail, the clinical perspective, what role do clinicians and doctors play in the Lilly Foundation or in the IMP as well?
So we work really closely with the medical professionals as well and the mitochondrial specialists, and I'm actually based in the Wellcome Centre for Mitochondrial Research up at Newcastle University. So kind of we work in partnership very closely with them, but with also the medical teams from the other highly specialised services. But this is something we certainly want to expand that, and we support patients and families from right across the UK, but we're always looking at ways of engaging with the medical professionals who may not have heard of the Lilly Foundation but do support mitochondrial families and attend medical conferences to raise awareness of the Lilly Foundation and the support that we can offer to patients and families.
Thank you very much.
Can I add something to that? I think worldwide, on a worldwide scale, we are very lucky as a mitochondrial community to have such good contacts with researchers and clinicians. I know it can be different in different medical sectors, but you know, clinicians and researchers really know where to find us, and they contact us and they invite us to participate and to take action on certain issues. So we are really lucky.
Great stuff.
I noticed that there's a question about informed consent in the context of mitochondrial disease, which is probably one that I should answer. The questioner asks, given the lack of therapy or cure for mitochondrial disease and the rarity of the disease, is the standard of informed consent different for patients with mitochondrial disease? Well, I suppose the simplest answer to that is no. The standard of informed consent is the same for any person participating in a clinical trial or research, or indeed for treatment. But I think what the question opens up is some of the issues that Kathy raised in her talk, that when you're dealing with a very complex condition and you're dealing with highly complex treatments, then there's even more need to pay careful attention to the standard of information that's provided for patients and potential participants. It's really important that people understand what they're committing to, for example, if they commit to a research project. And I think any way in which we can make that process better is going to work in favour of the science and work in favour of protecting and respecting patients' interests. So it's very important that patient organisations work responsibly. And I can see that Lyndsey wants to add something to this.
I was also just going to follow on from that and just say that we have worked very closely with the medical professionals again up in Newcastle around mitochondrial donation. And I know, Kathy, you mentioned that it's kind of one of the challenges is getting that information, kind of complex genetics and information across to patients or women or couples who are considering their reproductive options. And again, that's something we've done in our Lilly capacity, that we've worked with the clinical team up in Newcastle and helped develop an innovative clinical service. So that's the Mitochondrial Reproductive Advice Clinic, which is a clinic for women to come in and discuss their reproductive options from a very early age. So whenever they want to come in, they can discuss that with the medical teams and again get that quite complex information across so people can make informed decisions.
Is there anything you wanted to add to that, Kathy?
No, exactly. I think also, as you both said, like much has been made over the past year in terms of progress, and patients are much more direct access to this information, but it still can be quite overwhelming for them to have very often the information at a specific point at once, and not having like a follow-up session directly afterwards to kind of like debrief about that and ask their specific following up questions. So maybe more still can be done on that specific level.
Could I bring in Gemma or Sneha on that level about patient experience and research? Because from their work, I know that there's an added value for a design process that can sometimes be a real positive aspect of engaging patients.
Yes. Go for it, Sneha.
Okay, I'll give it a go and then you can build on it, hopefully. But I think something that listening to specifically the conversations in the first session about the scientific perspective and a lot of how you understand mitochondria and mitochondrial disease is something that stood out from our experience of working with patients, is that kind of translation of understanding a very medical condition from the perspective of someone living with it, in terms of how does that impact them on a day-to-day level. And for example, in some of the other long-term conditions that we worked on, like diabetes, it's more that from patients, the questions that often come are, how do we manage more in terms of our lifestyles, and how do we make sense of information in a way that we can use and how it's going to impact us on our everyday sort of life and quality. And I'm sure Gemma can come in and talk a bit more about that. But it's that sort of tangibility, I think, that the design process offers in terms of helping to make these connections more tangible and understandable and visual for people.
Yeah, I'm slightly conscious of the time, and I think that Kostas wanted to say a few concluding words.
Yes, I think we can just have one very final question to Gloria that has to do with statistics of how many women of Black, Asian and minority ethnic background are part of the Mito Women Initiative. And then after that, I will close the session. If you want to address that, Gloria?
Yes, this is something very interesting to do. We don't have numbers yet, but definitely this is something that we will work on in the future.
Okay. Thank you very much. So what I would like to do is then conclude the session here. And I would like to just thank a lot of people. So if I can just share my screen and give my thanks. to, of course, all the speakers, the Q&A moderators, my co-organizers, all the attendees. I think we were more than 300, which is quite impressive, I think. All the sponsors, the FRSE, the Lilly Foundation, University of Glasgow, the Glasgow School of Arts. In particular, I'd like to thank Hana and Sarah that behind the scenes have done a lot of work, and also in setting up the Zoom webinar and handling all the logistics, and the other teams at FRSE that helped with the promotion of the event, the comms and the PE and the fellowship team. You will receive, all the attendees will receive a feedback questionnaire with an email just at the end of this session, and this event will be broadcast on YouTube to increase dissemination. I'm sorry about the technical problem in the last session, but I think overall it was a very worthy discussion. I would like to close that now and thank all the participants. Thank you for your engagement. I hope this can improve mitochondria awareness. And it seems to me that this is something that we can probably think to organize again in the future, if people are willing to do so. So thanks again, and I will now close the event. Thank you for the participation, and I hope to see you soon. Thank you.